This is the short form of the population genetics lecture, covering only the most high-yield topics in the lecture, stressing what is most important for you to get out of the lecture. This slide is extremely important because it summarizes all of the mathematics that will be used in this lecture. The first is the most important formula, which basically says if you take the frequency of all of the dominant alleles P and add them to the frequency of all the recessive alleles Q, they equal 100% of all of the alleles. That makes sense anytime you have a dominant and a recessive, so a two-allele system. All of the individuals should have either the dominant or the recessive allele. Note that P is always the dominant and Q is always the recessive. So when we square this first equation, we get the second equation where P squared plus 2PQ equals Q squared. If you look over here in the Punnett square, you can see that basically all we did was we put the P and the Q on one axis and the P and the Q on the other axis for the square. And then when we get them together, we end up with square because we're saying P times P equals P squared. That P times Q equals PQ. And of course, the same happens here. So we end up with two of these PQs, one each of P squared and Q squared. They are equivalent, of course, to our homozygote dominant for P squared, carriers for PQ, and Q squared is our recessives. Likewise, you need to know when the Hardy-Weinberg equation is not valid. These two equations that we just went over are called the Hardy-Weinberg equation. They are not valid if random mating is not occurring or if there is a high mutation rate or some sort of a gene flow. Gene flow, remember, would be people entering and exiting the genetic pool. If no selection, you can have no selection against the phenotype for it to be in equilibrium, and you must have a relatively large population. We will go over all of these assumptions for the Hardy-Weinberg equation when we get to the second hour. They are very important, but questions about them will be more asking you, do you understand that this population is not randomly mating, or do you understand what gene flow is, or do you understand what mutation rates are? They are not going to be extremely complex questions for the most part with regard to determining whether a population is in equilibrium or not. Okay, on this slide, I want to remind you that prevalence and incidence for the purposes of population genetics are both considered to be synonymous, simply because we can't actually differentiate them at the population level particularly well. So we use both of them interchangeably. I know that that is not how you would do it in epidemiology, but that is how we do it in population genetics. Please remember that autosomal recessives have two copies of the allele. We usually say they are Q squared. In the case of someone who has an X-linked disease, it is typically represented by Q, and they will only have a single copy of Q if they are male. Please also remember that in dominant diseases, that the frequency of the dominant allele in the population will be both P squared plus 2PQ, because both the P squared individuals and the PQ individuals are both exhibiting the autosomal dominant phenotype. Remember that these are the frequencies, and this P squared is equivalent to big A, 2PQ is equivalent to big A, little a. Okay, there are 6 kinds of questions that can be asked to you about Hardy-Weinberg, all of which require a small amount of mathematics. So you need to have a calculator out to do these problems. Although you can do them in your head, it's probably a good idea to check your math, particularly on the exam, to make sure you know how to do them. Make sure you understand how to use the calculator on Examplify when you take the exam. Remember again that 2PQ is the frequency of heterozygotes. The frequency of heterozygotes within a dominant population would be 2PQ over P squared plus 2PQ. Remember that either P squared or Q squared is the frequency of the homozygotes. And remember, of course, p squared is for the dominant and q squared is traditionally for the recessive, and that we use only p or q in a hemizygotic inheritance. Please also note that there is a way to find the frequency of the allele in the population when the number of alleles is given. This is the math for it. We'll show you examples of how to do these kinds of problems. It looks a lot more complicated here than it actually is. It's simply saying the number of people who are heterozygote have one allele, and we add those number of alleles to the number of people who have homozygote alleles, but we multiply them by 2 because they have 2 copies of each allele, and then we divide them by the total population multiplied by 2. Although this is not a commonly used method, it is one that is tested on the USMLE. I have seen practice questions that deal with this type of question. And then lastly, the sixth kind of question would be one to calculate the risk from an ancestor from the oldest person affected. This is similar to the risk assessment that you've already done with Dr. Larson back in semester 1. But we're adding the component of population numbers in there as well. So we no longer will ignore the individual from the population that we don't actually have genetic information on. Now we add that in. It is important that you understand and can differentiate when the question is asking you for something that can happen versus when something must happen. So the term can in a question would be an either-or event, and that's when we add the risk of the father and the mother together to find the risk of the child. But if they must both occur, then we have to multiply them. You would use the can phenomenon when you're looking at the likelihood to be a carrier of a disease that is recessive or that a child will be affected by a dominant disease. Whereas you would use the must when we're looking for homozygosity in a recessive trait. Now in this lecture, I spent a significant amount of time, most of the first hour, teaching you how to work these problems. The important parts to note here in a problem is that if the problem tells you an incidence, this is equivalent to q squared if it is a recessive allele. If it is a dominant allele, it's just equivalent to p squared plus 2pq. In this case, we're talking about a child with facial deformities like cleft lip, which is a recessive allele, and so this would be q squared. This would also be q squared for the other parent. At first, we're looking for if they have an allele for this, then we would add it because they only have to get an allele from this parent or this parent. So we would add the 2 frequencies. And remember, just like you did in risk assessment, you take the frequency of the mother's risk times 1/2 to get to the child, and the father's risk times 1/2 to get to the child. And then we would add those together. And once we add those together, we get that there's a 14% chance that this child will be a carrier. We're going to repeat the process for the second question, which is what is the risk the child will have a cleft lip? And that would be doing exactly the same thing, except to be a child with cleft lip, they must have both an allele from their mother and an allele from their father. Therefore, we must multiply the 2 frequencies and then multiply by 1/2 as well. So this is 1/2 the mother's frequency times the father's frequency times 1/2, which gives us a 0.074% chance that the child will have cleft lip. So here we have 4 people who have the disease beta thalassemia and 171,500 carriers. These people have 2 alleles because they have beta thalassemia, and you will remember that beta thalassemia is a recessive, autosomal recessive trait, and these people have 1 allele. So the population here will be 68,600,000. And we have one allele for each heterozygote and 2 alleles for each homozygote, which gives us a grand total of 171,508 affected alleles. And we simply take this number of affected alleles and divide by the total number of alleles in the population to get our allelic frequency. And in this case, that is 0.25%. So since this says it is the allele, HBS is the allele for sickle cell, that tells us that this is the number of alleles in the population, not the number of affected people with sickle cell. If they meant sickle cell people, they would have to say the incidence of sickle cell. So that tells us that that is Q by simply dividing those numbers. And we do the same math that we've done in the previous problems to figure out the risk for a child to have this disease. So remember that each time you pass from one generation to the next, you must multiply by 1/2. So as we multiply by 1/2 for both of these parents, we get this number times 1/2 times this number times 1/2 to figure out if the child will get sickle cell. And to figure out if the child will have just one allele, we would add them instead of multiplying them, and we would get a 34% chance as opposed to a 2.5% chance. In a case where you have a disease that is very devastating, we typically always diagnose the patient first before we do any genetic testing. In a disease like Huntington's disease, it's very easy to do the diagnostic testing for this patient. So as soon as this person came into the office, we would have drawn blood and looked at their repeat number for Huntington's disease. Remember, Huntington's is a trinucleotide repeat disease, and the more repeats you have, the more likely you are to develop Huntington's disease. And since her brother, who is 2 years older, has been diagnosed, she has a high chance of carrying the disease, so we would initially always test her. Since we don't know anything about her husband, we must calculate for the husband. The incidence of Huntington's is actually 1 in 15,000 approximately, and that would of course be, because it is a dominant allele, both PQ plus 2PQ. Now there's an interesting trick that we do to figure out 2PQ here, because we know that Q squared has to be essentially 1, because this is very, very small. Because we know that that's essentially 1, we can make an assumption that P squared must be even smaller and that it must be essentially 0, and we ignore P, but basically we just use the frequency as the 2PQ in the problem. So we put in the incidence here as 2PQ for the father, multiplied by 1/2, and we get 1 in 30,000 as a chance for the child. Okay, the next major concept I needed to go over with you was the concept of not in equilibrium. When a population is not in equilibrium, we cannot use the Hardy-Weinberg. And this is just an example of that phenomenon. Come out with something that still doesn't look anything like our population. It allows us to get the number that's in the middle for the heterozygotes, but we don't ever get, no matter what numbers we try, we would never get all 3 numbers or anything close to them, which tells us this population is not in equilibrium. That means we cannot do population genetics for this phenotype, this genotype, on the Bayesian population or the University of Florida population. Now, why is that? That's probably because the population is not in equilibrium, and here are the reasons that populations are not in equilibrium most often. When we fail to meet Hardy-Weinberg, we cannot use this type of analysis to study this population. Now let's go into some of those traits that are very important. One of those is random mating. Random mating is normally assortive, We don't actually truly randomly mate in humans. We pick our mates because we have characteristics that we think are compatible with us. We pick people who are like ourselves or are very compatible to ourselves. That means it's not truly random, but in general it works out to be equivalent to random most of the time. Okay, what is the founder effect? The founder effect happens when we have a very small population and one member of the population or a few people in the population have a trait, but because of the random mating within that population, that gene becomes more and more prevalent than it ever was before. That is called a founder effect. Founder effects usually happen only in isolated communities. When we say isolated, we mean sexually isolated. That means they do not outbreed. It's generally due to a gene flow event as well. And the frequency of consanguineous relationships is generally very high in these populations because they are very small at the beginning. And that helps to further enrich for these rare diseases. Generally, we consider the mutation rate 0. It's not truly 0, but it's so small that we ignore it. In most cases, it becomes fixed, and it turns out that it is balanced by other factors like selection rates. This tells us that then we need to be very careful when we look at our populations, and we need to look at what matches our population. Not always should we just use the general data. If I have a young Southeast Asian couple, I would get flawed results if I'm comparing them to the whole U.S. population. The effect of inbreeding is extremely important to understand because it raises childhood mortality rates, because closely related individuals enrich for homozygous genes. This means that autosomal recessive traits can be enriched for in consanguineous relationships. Spontaneous mutations are very high in older parents, but there's a difference in the kinds of spontaneous mutations depending upon which parent is giving the mutation. In men, the most common type of mutation that accumulates and they pass on to their children are point mutations, and in women they are recombinatory errors. Next, I'd like to remind you about gene flow and gene drift and just differentiate these to you. Gene flow is where individuals come into and out of the population, changing the frequency of the alleles in the population by taking alleles into or out of the population. Whereas genetic drift is change in the genetic makeup over time without a change in the population. We also, lastly, need to know about selection pressures that alter the gene frequencies. Selection pressures alter by virtue of genetic drift, not gene flow. They do so by either selecting for or against specific alleles. And occasionally there's actually no selection or a neutral selection. Those are much harder to fix in the population. Do remember that it's easier to get a recessive allele that becomes common in the population than it is a dominant allele, because if the dominant allele is lethal, then after a single generation, everyone dies who has it, so they don't tend to pass it on. Whereas if you have a recessive allele, all the heterozygotes can actually survive just fine. Selection comes in 3 types: stabilizing selection, directional selection, and disruptive selection. These basically just say what is happening to the population, and we'll now walk through a few of these. For stabilizing, it is a beautiful example of stabilizing selection is in blood pressure, where it is not particularly good to have high blood pressure because you increase your likelihood of stroke if your blood pressure goes high. Likewise, you have a tendency to faint Fainting people reproduce less, so this is also a negative toward negative selection against people who have low blood pressure. And so over time, the peak of the normal distribution of people's blood pressure has gotten smaller and smaller across the human population. An example of directional selection has been the evolution of humans. Lactose intolerance, which is the ability to continue to consume milk into adult life, as you will recall from Digestive 1. The most common example of disrupted selection is Darwin's finches. A medically relevant version of that is human pigmentation, a single point mutation at amino acid 111 in melanin is what allows for people who were closer to the poles, the North and South Pole, to develop lighter and lighter skin. Selection. So we have the people near the poles have a very different skin color than the people who are equatorial, where dark skin helps to protect you from getting melanoma. Lastly, we don't worry about most of these issues because selection and mutation are typically well balanced. I told you before that mutation is typically close to zero, but not exactly zero. It turns out that it exactly balances in most populations with the selection, and so we get to let them negate each other, and we don't have to take them very seriously. But we always have to check, as the example we started the second hour with showed us. If you want to be very precise, you do something called a chi-square analysis. But in general, for all of the USMLE questions, you'll be able to do it the way we did at the beginning of this lecture. Just look at the numbers and say they don't make sense relative to what I know the frequency should be for a normal distribution. And that's good enough to tell you it's not in equilibrium. Okay, so at this point you should be able to practice problems using the Hardy-Weinberg equation, know how to read a vignette and figure out what it's telling you and what you need to do with it. You should be able to differentiate incidence to prevalence, but know that in the case of population genetics, we use them synonymously. You should be able to figure out the frequency of an individual homozygous allele or heterozygosity or the trait of a child or a grandchild using population numbers. You should know the limitations of population genetics and how to recognize them. You should be able to explain inbreeding coefficients and the effects of inbreeding, as well as know what are the assumptions made in the Hardy-Weinberg equation and what they mean.